So the next speaker is a star, and you all know him. He's been in this field since the beginning of time. I trained with Zev Rosenwax and Jacques Cohen, and basically is when he and the late great famous, whose name I'm blanking, Yuri Verlinsky, are some of the first to be doing PGS, and he's been with it through thick and thin, and he's going to show us some big numbers and some nice data. Santi?
Well, it's difficult to follow this great talk from Mark Hughes and now have to talk about the same thing. First, thank you to John for inviting me to this great meeting another year. I'll try to rehearse in different ways the talk of Mark Hughes, and mostly I will focus on PGS. So, as you know, and you've seen this many times, there is an increase of chromosome abnormalities with advancing maternal age, as low as 30% in young patients, and it goes up to 80-90% in older patients. And because now, because couples are pursuing career and education, there is a delay in achieving pregnancies, and this basically is resulting in more infertility because of this, because there is an increase in aneuploidy with advancing maternal age. So you could say that delayed reproduction basically is infertility by aneuploidy, and we need to conscience our patients that they should either do fertility preservation or think about this seriously. The problem, therefore, and that's why we want to do PGS, it's because about 30 to 90% of these embryos will be chromosomally abnormal. These embryos seldom implant or miscarry. And now that we have these new technologies, PGS, what we call version 2, which consists of biopsying blastocysts. Blastocysts are, once we biopsy it, the biopsy is not detrimental. With these new technologies, chromosome comprehensive analysis, all the techniques that Mark has explained before, we have a very low error rate, at least when the embryo is fully euploid or fully aneuploid. There's only one study showing that blastocyst biopsy is not detrimental so far. I think we need a few more, and I'll show you also some data that actually it can be, depending how you do it. But basically, if you do it with the standard methods, it's not detrimental. So once you put all these things together, we really can avoid transferring aneuploid embryos, and then these results, at least in 3 randomized trials, in improvement in ongoing pregnancy rates. And in addition, it eliminates the effect of maternal age on implantation. The advantages of blastocysts are obvious. When we biopsy, we are sampling the trophectoderm, which is going to result in the placenta. We're not touching the inner cell mass. We have more DNA. Instead of 1 cell, we're biopsying several cells. We recommend about 5 cells, and I'll show you why. There are less mosaics, but still quite a bit of mosaics in the trophectoderm, but significantly less than on day 3, because some embryos that are complex abnormal and so forth, they don't make it to blastocyst. It reduces the impact of embryo biopsy, and that's the study by Scott that he published in 2013. And it's probably because of this we're not touching the inner cell mass. And also we have less embryos to process. Now that we are doing arrays and next-gen sequencing, each sample has an extra cost. So by only testing the embryos that at the end you're going to transfer, you are reducing the cost of the technique. And now that you have the best embryos morphologically and genetically, there is no reason to transfer more than 2 embryos. So it facilitates a single embryo transfer. But with every technique, you also can do some damage. So we have this paper that we have submitted recently, and it shows that if you biopsy more than 10 cells, actually you can decrease implantation potential of these embryos. So that's why we recommend only biopsying about 5 or less than 10 cells if possible. This was because one of the labs, I think it was Natera, recommended to this group, it's a center in South America, to send them more than 10 cells per biopsy. But they were doing also RACE-GH at the time, so they compare RACE-GH results with this center, and as you could see, if you biopsy more than 10 cells, then you have you could do some damage. Over the years now, I think that we have, for 2016, I don't think we have any single day 3 biopsies. The whole field has changed, basically, at least in the US, to blastocyst biopsy. And as Mark Hughes reviewed, there are several techniques that you could use, but the best one, the one that has the more resolution, it's high-resolution next-gen sequencing. And depending how do you, which of these techniques you use, you will classify differently up to 20% of embryos. So this is significant because it will have an impact on miscarriage rates. I will now focus again a little bit more on Mosaicism, which is what we can detect with next-gen sequencing better than any other techniques. Mosaicism has been well described many, many years ago when we were doing FISH and analyzing all the cells of the embryo for reasons of knowing what these embryos, why they stop developing, etc. Thousands of embryos were analyzed, all the cells, and we know that mosaicism existed a long time ago. About 30% of these embryos were mosaic at the time. But when we transitioned from FISH to quantitative PCR, RACE-GH, etc., those techniques could not detect it. So for the last 5-10 years, mosaicism basically disappeared from the vocabulary. But it's not that something that now we have invented with next-gen sequencing. With next-gen sequencing, what we see, and this is going to answer a previous question, basically, there is no increase of mosaicism with maternal age. It goes from about 16% in egg donors to about 11% in over 42. Actually, it looks like it's decreasing with advanced maternal age. But this is only because some mosaics will have full aneuploidy, so they are classified in the below row. And for aneuploidy, you see the typical increase in chromosome abnormalities with advancing maternal age. Actually, the more or the higher the maternal age, the more complex these abnormalities become. So, for instance, having more than 2 chromosomes involved, it's only 6% of embryos in egg donors, but it goes to 40% in patients over 42. This is another way of looking at this. Only aneuploid embryos increase with maternal age, while mosaics basically are stable. This is also showing you the same data, but depending on the cohort size. And what we've seen is that although chromosome abnormalities increase with maternal age, they are not correlated with the cohort size. So patients that produce few embryos more or less have the same rate of chromosome abnormalities of those that produce lots of embryos. This is important when you consult your patient because obviously the more embryos you produce, the higher the chance that you'll find at least one normal embryo. What are the clinical outcomes of when we have mosaics and we transfer them? Well, we started this a few years ago when we had a misdiagnosis in which an embryo that was diagnosed as normal by RACE-GH miscarried. It was the trisomy 16. And then what we did is reanalyze the DNA that we had stored. We stored DNA from all of our embryos. And by RCGH, it showed exactly the same result. It was normal, but then we did next-gen sequencing, and it shows us that it was a mosaic trisomy 16. So then we reanalyzed all the miscarriages that we had. I think it's something like 52 out of 25,000 samples or so. And what we found is that half of all the miscarriages that we have were, in fact, mosaics by next-gen sequencing. So just by going from RCGH to next-gen sequencing, we could cut in half the rate of miscarriages, which was already reduced from not doing anything. So if you don't do PGS, you would get between 17 to 40% miscarriage, depending on maternal age. With the RCGH, we have about 13%, and with next-gen sequencing, now we have about 6%. This, we thought, transferring mosaics. Obviously, if you transfer mosaics, you have a higher miscarriage rate. Also, mosaics implant-less. So this is data by Fraguli that was presented at PGIS in 2016. So they miscarry more, they implant less. But as you know, some make babies. This is the paper from Greco et al. And Fiorentino. This was, their paper was transferring mosaic embryos found by RCGH. That's only about 4% of embryos can be detected by RCGH as mosaic, compared to about 20% with next-gen sequencing. But still, in the cases in which they transfer it, some of them implanted. Which of these mosaics have a higher chance of implanting? So we look at it in different ways, by the percentage of abnormal cells and also by the type of mosaic. So if we have, for instance, mosaics in which more than 2 chromosomes were involved, so you have monosomy 21, mosaic trisomy 13, etc., 3 or more, we call them complex mosaics. And they seldom implant. Even they seldom miscarry because probably they don't even implant. Then we have mosaic aneuploid, in which, let's say, 1 or 2 chromosomes are in mosaic form. And then depending on the percentage of abnormal cells, so 40% or more or 40% or less, you see that the ones that have more abnormal cells, they implant less and miscarry more. And the ones that have more normal cells, they implant moderately well, 46%, and they have 11% miscarriage rate. So still below normal, but obviously if you don't have anything else, it's worth transferring these embryos. And then the ones that do best are the ones that have partial aneuploidy. So there is only one piece of a chromosome that is missing or extra, and it's in mosaic form. So some cells are normal, and some are missing or have an extra piece of a chromosome. And those 55% are ongoing or so. So pretty close to euploid embryos. This is data from NYU, in which they've done a lot of cases with both techniques, RCGH and next-gen sequencing, and they already you already can see a significant improvement in pregnancy rates, ongoing pregnancy rates, by switching to next-gen sequencing and lower miscarriage rate. So what we are proposing is that we switch how we are transferring embryos and classify embryos in 3 categories instead of 2. Before, we were classifying them as normal or abnormal. There was an intrinsic error rate in doing that because of mosaics, between 2 and 10%, depending on the technique. You have false positive and false negatives, and then you have the situation like Gleicher, in which if you transfer abnormal ones, some of them may be normal or mosaic and still produce babies. What we're proposing is to classify embryos now into 3 categories, normal, mosaic, and abnormal. The normal ones will be really normal, the abnormal ones will be really abnormal, and then you have this gray zone in between, in which you will have higher miscarriage rate, lower implantation rates, but still some of them can make babies. And we propose this new paradigm in this paper with Jamie Griffo and Dagan Wells. Again, so before with the RACGH and the other techniques, about 60% of embryos will be normal and about 40% will be abnormal. Now we're going to classify, and that's something that some doctors are complaining that, wow, we have less normal embryos now. Yes, but because now we're defining a zone, this gray zone of mosaics, in which if you transfer them, although before maybe we would have classified them as normal, they have higher chances of miscarrying, and we did have some misdiagnosis when we transferred these normal embryos because they were in fact mosaics. And same thing with abnormal ones. Now we have also fewer abnormal ones because some are classified as mosaic, and these have some potential to implant. So I think that this is a more precise way of classifying these embryos, and with time, probably we'll be able to even give you more details about these mosaics and which to transfer and which not to transfer. Right now, very few of them have been transferred. So the data that I showed you before, depending on the type of mosaic and the percentage of mosaicism, it's based on this little data that we have, but it's going to get more and more precise, and probably will be able to give you better ways to counsel your patients. So in summary, for mosaics, nature sequencing is the only method that can detect most of them. About 21% of mosaics of embryos are mosaic, although some of them also have other abnormalities, in which, in that case, you wouldn't transfer them. They miscarry more, so if you have enough embryos that you have euploid embryos and you don't transfer these mosaic embryos, then you have much better results. They implant less, so same thing. If you have euploid embryos, first transfer the euploid ones. But about 40% of them, and these are data that we did biopsying different areas of the same blastocyst, 40% of these mosaic embryos will have normal inner cell masses. So this is more or less about the same as if you don't do any diagnosis. So the rate of mosaicism or the percentage of normal inner cell masses will be more or less the same as an undiagnosed embryo, which about 40% of them will be normal at let's say, above 35. So you have to consider them to transfer them if you don't have better embryos. Because they will make babies. Now, if you decide to transfer them, we totally recommend, at least for now, to do prenatal diagnosis with amnio. If you do CBS or NIPT, you're testing again the same tissue, the trophectoderm. So it's not going to give you much more information. The overall results of PGS, now, as you know, there have been at least 3 randomized trials that all have shown significant improvement in ongoing pregnancy rates. The caveat or the criticism of these studies that all of them are relatively small. All of them have been done in good prognosis patients, so patients that produce less chromosome abnormalities than patients of advanced maternal age. In a way, it's even more encouraging, but that's one of the criticisms. All the meta-analyses that have been done and a couple of other papers all agree that PGS improve results. But there's still people asking for more randomized trials, and more randomized trials are coming, as I will show you. This is another study that has won this year the surprise paper, comparing fresh and frozen transfers, and basically, if you consider the day 5 embryos, the embryos that arrive to blastocysts on day 5, basically there is not much difference. The problem is on the embryos that reach blastocysts on day 6. Those obviously do much poorer in fresh transfers. So when you put all the data together, and this is our data from reprogenetics, but also this has been seen by Richard Scott in his data, by Genesis in his data, and later show you other data. Basically, if you transfer a euploid embryo, they implant equally well at any age. That's obviously not what you see when you don't do PGS. So basically, the biggest reason for the decrease in implantation potential with the vast maternal age is aneuploidy, and now we have basically solved that problem. So this is data that has been collected by David McCullough from NYU, analyzing the SAR data from 2014, and he finds exactly the same thing. Because now this is newer data. This was already data using newer technologies. You don't see this in previous years, but 2014 probably is the year in which everybody was doing PGS already with advanced technologies. And as you could see again, there is no decrease in implantation potential, at least up to 42 years of age when you do PGS, but there is obviously without doing PGS. Same thing with live birth, and this again data from reprogenetics. There is no increase in miscarriage rate when you do PGS. There is obviously a huge increase without PGS, and same thing found by David. No increase in miscarriage rate, at least up to 42, and a huge increase if you don't do PGS. Regarding live birth per retrieval, he sees a significant improvement up to above 38, and not much in the younger patients. But you have to consider that in PGS they had only one embryo transfer. Meanwhile, on the untested group, they have more than one. So this is similar to what Foreman found in their clinical randomized trial. They found that if you transfer one euploid embryo, it's more or less as transferring two untested embryos. And by just transferring one embryo, he also found that there is less premature deliveries on the PGS group than in the non-PGS group in the SAR data. Still, this doesn't answer the question that probably Gleicher and some people in Netherlands would say that what about cumulative pregnancy rates and what's the intention to treat? So he has developed this model that he presented at Cogent a couple of weeks ago. Basically, let's assume it's a patient with 13 embryos, and you want to transfer all these 13 embryos one by one. Who's going to win, PGS or non-PGS? So he made some assumptions based on the paper of Scott et al. About what's going to be the live birth rate. And basically, if you, let's say, you have 4 euploid embryos in that cohort, obviously you'll get to pregnancy sooner with PGS than with no testing, but at the end, if you transfer all the embryos, you would think that not doing PGS is better, because at the end you are going to transfer those 4 embryos anyway, and you could assume that there is a small error rate in the PGS, so therefore not testing these embryos will win over the time. However, if you transfer them, if you transfer first the euploid embryos and then you transfer the other ones, if that's something that you would consider, I wouldn't, then you reach more or less the same pregnancy rate, although obviously you get to pregnancy sooner with the PGS. However, what the model that the people arguing for transferring one embryo at a time and transferring all of them, they don't consider is that there is a dropout in treatment. Here he has modeled only 7.5 dropout, but I think there is a paper by the group Boston IVF that it's close to 20%. So once a patient fails a cycle, and especially if they miscarry, the dropout is really considerable, close to 20%. But just modeling a 7.5 dropout, PGS wins all the time. In both ways, if only you transfer only the euploid embryos, or if you want to transfer all of the embryos starting with the euploid ones. So I think that's another argument that obviously this, I don't know if you need to do this in real life. I think that there are enough evidence that PGS works, but if somebody wants to do it, they have to consider that dropout, and then at the end of the day, I think that PGS will win. There is a study also that I want to comment. There was a study recently. It's not a randomized trial, but it's a study in which they compare patients with recurrent pregnancy loss doing nothing or doing PGS. Sorry, the age here has moved. So basically, the age of the IVF plus PGS and IVF no PGS was 37. Meanwhile, in the control was 35. So already there is something wrong here. 2 years of difference is quite a bit. But if you look at the pregnancy rate in the patients that had IVF plus PGS, it was significantly higher than in the control group. Miscarriage rate was significantly lower. Live birth rate per transfer was significantly higher with PGS. The thing is that they mix IVF plus PGS and IVF without PGS, and the middle group was cycles that they cancel. The intention to treat was PGS, but then they canceled because they didn't have enough embryos, according to them, to do PGS. So if you do PGS, obviously it's better than not doing PGS. And to answer the critics that there are not enough randomized trials, this trial is called the STAR trial. It's already finished. It has randomized 300 patients in the control group and 300 patients in the test group. It involves 9 PGS centers, 34 clinics in 4 countries. And it's the patients are moderately of moderate prognosis. So you have a patient from 25 to 40 with 2 blastocysts or more. Some of the randomized trials that have been done, patients had on average 9, 10 blastocysts. So this is a moderate group of patients. And I think this will, solve most of the doubts of all the critics that are still waiting for more data. Obviously, I cannot say if this is going to be positive or not. The trend, because all the other studies that have been done, I guess it will be positive, but we won't know until the data is analyzed, and this is not going to happen until December of this year. And we hope to present this in ESHRE next year. Interesting thing we have found, and Mark Hughes also presented the same, there are differences in chromosome abnormalities depending on the IVF center. Obviously, you could say, well, my population is much different than yours. So we thought about how to compare these different centers, and I think that the best group is egg donors. Egg donors are usually chosen everywhere in the same way. There are some guidelines. All of them are young, fertile, etc. So it's a good group to compare center to center. We've done extensive statistical analysis to weed out confounding factors. So the egg donor age, infertility reasons, day of biopsy, paternal age, other indications such as male factors, sperm donation, cohort size. And the only confounder factor left was the center itself. So then we compared these centers. We've done 1,600 PGS for egg donation. From 42 centers that at least had 10 egg donation cycles. Average age was 25, and the average aneuploidy rate was 31%. This was data with raised CGH, so here we were still not distinguishing mosaics from aneuploidy. So the average chromosome aneuploidy rate was 31%, but it ranged from 18% to 60%. So huge difference between centers. Also now, more recent data, we have seen the same, still a huge difference between centers, but also in different types of chromosome abnormalities. So some centers have high aneuploidy rate but low mosaicism rate, and others have the contrary. We have two pieces of evidence, I think, that point to different reasons for this. One, we had several centers that had enough cases that we could compare doctors within the same center. And we found in some, so we asked them, all the doctors in this center stimulate in the same way and treat the patient in the same way. And for instance, one center that has many doctors, there is only one doctor doing hormonal stimulation. So we didn't consider that center. Actually, we did, but we didn't see differences in this type of centers. But in the centers that they said that each doctor stimulates differently, we did find significant differences in a couple of centers, depending on the doctor. So same IBS center, but different doctors, different chromosome abnormalities. Also, there is another piece of evidence. This is going to be presented at ISRM. And this is just comparing different media, same center, same doctor, two different media, and it showed different rates of chromosome abnormalities. So obviously there are different reasons why this could happen. We thought about hormone stimulation, although we look at hormone stimulation forever, and it's really difficult to pinpoint how this could happen. But it seems by the fact that some doctors may produce more chromosome abnormalities, that is the first thing that you would think. Although it could be which follicles they aspirate. We know that smaller follicles have more chromosome abnormalities than larger follicles. That could be just one difference and nothing to do with stimulation. It could be other things that they do. Definitely the laboratories, we know for a long time that changes in pH, temperature, etc., or even culture media could have an impact. But I think that most of the laboratory conditions, what they are going to do is to produce mosaicism. The good thing of this is that if we can create chromosome abnormalities, we can decrease them. So I think that PGS could be used also as a quality control for the IVF lab. And once you want to change one thing, do PGS and see if the chromosome abnormality rate, especially in mosaics, goes up and down. And it could be a good way of seeing if this thing that you want to change, it's a good change or a bad change. Just a couple of slides on mitograde. We talked quite a bit about chromosome abnormalities, but also what we've seen, this is data mostly from Elpida Fraguli, is that in addition of the chromosomes, mitochondria also have an impact on embryo viability. As you know, mitochondria are inherited completely from the egg. And there is a bottleneck at a certain point in which some embryos will have more mitochondria than others. Some mitochondria will divide, some will not. So you may have different populations of mitochondria at the beginning and at the end a different ratio of them. This is important when you have mitochondrial disease. But what we've seen is that some embryos produce much more mitochondria than others, and above a certain threshold, these embryos never implant. You could analyze this in different ways. So what we use is a threshold, and basically we classify embryos below the threshold as normal amounts, and above it as abnormal and non-implanting. Other centers, what they do is to classify them in quartiles and give a different percentage of implantation depending on the quartile. What we've seen is about 15% of euploid embryos, they never implant because they have this problem. They produce too many mitochondria. We've seen that this is not associated with morphology. It's only mildly associated with aneuploidy and paternal age. And so far, we haven't seen a correlation with mutations. But still, we have no idea why this is occurring. Obviously, we assume that it's something is wrong with the mitochondria, and the egg is producing more mitochondria to compensate for that. But right now, we haven't found out yet what's the problem with those mitochondria. What we're seeing, as I said, is that these embryos never implant. It's not very common, so we have only about 280 transfers now with follow-up. 200 of them where the mitochondrial levels were normal, and 33 of them were abnormal, and none of these abnormal ones implanted, although they were blastocyst and they were euploid. So, in summary, as I said, about 30 to 90% of embryos are aneuploid and they never implant or they miscarry. And now that we have these wonderful techniques, we've seen that if we choose euploid embryos, not mosaics, then the decrease of implantation with advanced maternal age, basically it gets eliminated, at least up to age of 42, and this has been seen now by all the PGD labs, and also the START data shows the same. And at least in these 3 randomized trials, and hopefully the START trial will show the same, there is an increase in ongoing pregnancy rates when you do this, when you do PGS. And now that we have next-gen sequencing, we can even reduce the false positives and false negatives that we had before, because now we know where they come from, and if you don't transfer these mosaic embryos, you will have higher implantation rates and lower miscarriage rates. And if you don't have anything, you don't have euploid embryos, then you could transfer them and at least have a small chance of having a pregnancy, not a small, but reduced, and not throw away embryos with some potential to implant. Thank you.